Hello all viewers and welcome to Instant Biology by Dr. Neelab. The topic that we would be dealing today is recombinant DNA technology. Okay, so although I know that recombinant DNA technology is a vast field and it cannot be completed in one lecture, but we would try and give you an overview of this recombinant DNA technology. Okay, so recombinant DNA technology is abbreviated as RDT. Okay, so this is the basic or the backbone of genetic engineering. This is RDT is the backbone of genetic engineering, and mostly in biotechnology also. In biotechnology it is mostly employed in biotechnology field, so as to what do you say so as to utilize the microorganisms in such a way that they are beneficial for human beings. So let us write this down, that utilization of microorganisms in such a way that they can be useful for human beings, right? So this is basically the role of recombinant DNA technology. Now let us understand what recombinant DNA technology is. What does it really mean? Let us understand this. Okay, so by recombination, what do we mean? It is recombination means the joining of two DNA molecules having different origin. Okay, so it is the joining of two DNA molecules that have different origins. So let us understand this by means of a diagram. Suppose this is a DNA molecule from bacteria. Suppose this is a DNA molecule from E. coli. Let us write this as E. coli. This is the DNA from E. coli, and we are joining a piece of DNA or a fragment of DNA from human beings. Okay, so this is the fragment from human beings. Okay, so what is this in completion called? What is this entire molecule called as? It is a hybrid. It is a hybrid between bacterial DNA plus human DNA, and therefore it is called as a recombinant DNA. Recombinant DNA. So this is what we meant by saying the recombinant DNA. It is a combination between the molecules or DNA molecules from having two different origin, right? So this was about the what is recombinant, what does recombinant mean, right? So let us understand this better with the help of an example. Do you know that how insulin from the human beings for the human beings basically is made? Insulin is cloned into the E. coli, and then it is utilized to express or to prepare or to manufacture the protein insulin, and then only this insulin is obtained from industry in the form of medicine, and this is utilized for patients having diabetes. So recombinant DNA technology has played a great role in the manufacturing, in the preparation of the insulin. Previously it was used using traditional methods, right? So this was about the overview of recombinant DNA technology. Now let us understand step by step that how this recombinant DNA technology works. Okay, so how does DNA recombination technology works? Let us understand this in a stepwise process. First step that we need to understand, that we need to study, is identification and isolation of gene of interest. Okay, let me write it down. Identification and isolation of gene of interest, right? So what is the gene of interest? In our case, we are saying that we have to work upon insulin. So insulin would be the gene of interest for us. How do we identify it? We, of course, we know that we want to work upon insulin, so insulin would be the gene of interest for us, right? So insulin would be the gene of interest for us, definitely. Now we want to take this insulin from somewhere. What would be the source of insulin? From where can we get this insulin? We can get this insulin from three places, right? Firstly, the first place that we can go on to search the insulin would be genomic libraries. The second one would be cDNA libraries, and the third one would be the chemical synthesis. Now what do you understand by genomic library? Genomic library comprises of the entire genomic content of a particular organism that has been maintained in the form of clones, that have been maintained in the form of different clones, each clone having a different fragment of the genomic content of a particular organism. What is cDNA library? The cDNA basically is different from the genomic DNA because the cDNA does not comprise of introns, okay? Only exons are present. That is cDNA library. And chemically also, synthesis of the particular DNA that you need can be synthesized now with the advancement of the technology, right? So these are the three methods that you can utilize for identification and isolation of your gene of interest. So the gene of interest that we want to work on is insulin, right? Okay, so this is what we wanted to work upon. Now let us go on to the second step. The second step is construction of recombinant DNA. Construction of recombinant DNA, rDNA. So how do you construct the recombinant DNA? This is the main topic that we have to work upon. Let us take a different page. Right. So how do you construct a recombinant DNA? In construction of a recombinant DNA, first of all, you have to add your DNA to a vector. What is a vector now? Vector is a vehicle that can carry your DNA. Vector has some specific properties, such as it should have an ORI region, it should have the ampicillin resistance or the antibiotic resistance gene. We would talk about this in detail in the upcoming videos. Now first of all, we need to understand that how can we insert our gene of interest into the vector. Okay, so suppose this is a plasmid. Okay, we have obtained this plasmid from E. coli. This is a plasmid. It is complete. Okay, this is a plasmid. Now what do? This plasmid can be cut off at places with the help of restriction endonuclease enzymes, right? You can cut this plasmid at different places using restriction endonuclease enzymes. Now after cutting, what will happen? This would look like something like this, because the enzymes, which type of enzymes can be used over here? Enzymes such as EcoR1, BAM, H1, etc. Such enzymes can be used to cut this plasmid, right? Now when you obtain this plasmid, it does not have this particular region because you have cut this off. Now you can add your gene of interest to this particular region, right? So how will this look like when you add up your gene of interest? So you can add your gene of interest over here. So this is the gene that would code for insulin. Humans, right? You have to prepare the insulin for humans. So this was the vector. or the plasmid of E. coli, and this was the insulin from humans. So this is how you prepare your recombinant DNA. This structure is called as recombinant DNA. Okay, so this is formed by ligation of this human insulin gene to the vector DNA, right? This was formed by ligation of these two. Now coming to the third point, the third point that we need to work upon is the introduction of recombinant DNA into suitable organism. So let me write this down. Third point is introduction of recombinant DNA into a suitable organism, Okay, so let us understand this. The vector that you have created, the hybrid that you have created over here, would not or cannot multiply by its own. The thing that we need is multiple copies or increased number of copies, high number of copies of this particular insulin gene that we are working upon, that we need to have it. So we need multiple copies of this, but until and unless you place this vector, this entire structure that you have obtained in a living organism such as a bacteria, this won't multiply. Okay, so the next thing that we should be aiming or we should be working upon is the introduction of this recombinant DNA into a suitable organism. So in our case, let us consider this to be E. coli, right? So let me draw this. So this was the recombinant DNA, this was the original plasmid, and next what did we have is we had this, our gene of interest, right? Now what do we have to do is we have to introduce this to a E. coli, right? Suppose this is the E. coli. Suppose this is the E. coli cell. It won't be in a rectangular shape, but just for your understanding purpose I'm showing this. This is a E. coli. You need to transfer this cell into in here, right? So how would you transfer? That is also a thing that we should know about. There are multiple methods to transfer. Let us understand them. First one is the physical method. Second one is the chemical method. And finally there are virus mediated. So in the physical methods like electroporation can be used, methods like liposome mediated transfer can be used. In chemical methods like PEG can be used, and CaCl2 methods can be used, and finally the virus mediated method. So with this, what will happen is your recombinant DNA, that is this, would be transferred into E. coli, that is this, right? So it would get transferred inside this. Now we have to work on the next point. What would be the next point? You are not at all sure that whether this particular your gene of interest or the recombinant DNA that you had produced over here, did it transfer into this E. coli? You are not sure of that, and that cannot happen or does not happen fully, or 100% transfer of recombinant DNA into the suitable organism does not take place. So we have to screen. So the fourth point is screening of the fourth point is selection of transformed cell, not screening. Screening would be the next step. So this would be selection of transformed cells So what are the transformed cells? Transformed cells would be those that have taken in the recombinant DNA. So all the cells, all the E. coli cells would not take up the recombinant DNA. Only some of the cells would take up the recombinant DNA, and we have to select those cells. Okay, so let us understand, or let us take up some of the cases. What can happen when you are transferring this rDNA into the E. coli? What can happen? There are three possibilities basically. First possibility is non-transformed cells. Non-transformed cells would be the first. So in case of non-transformed cells, what will happen? This is the E. coli, and it does not have any what do you say, any recombinant DNA. Okay, so this is the first one. Coming to the second one. Yeah, so second one is transformed cells, but non-recombinant vector. Transformed but non-recombinant vector. So the cells have been transformed, but the vector is the non-recombinant one. Let us understand this. Okay, so this is the E. coli cell. It has been transformed, but the cell has been transformed by the non-recombinant. What does non-recombinant means? Non-recombinant means that only the plasmid portion has been entered into this cell. Only the plasmid portion, not human portion. Human portion has not entered into this, which means that recombination also is not a 100% process. The recombination formation efficiency is also not 100%. Okay, so these cells, these E. coli cells have been transformed, but they have been transformed by non-recombinant vector, right? So coming to the third point, what is the third possibility that can happen? Transformed with recombinant vector. So this is the population that we want, transformed with recombinant vector. So this is the population that we want to work upon, that we are interested in, transformed with recombinant vector. How would this look like? This is the E. coli cell, right? And it has the plasmid as well as the gene of interest that we had put into it. Okay, so these E. coli cells have been transformed by recombinant vector, right? So this is what we were aiming for. Now we have to select that. How can we select amongst these three? I have shown you the possibilities. These three were the possibilities. We need to select or screen this. The third part we have to screen, right? So whether the transformation has taken place or not can be screened by two methods generally. Okay, so let us understand this. The transformation has taken place or not can be understood by three methods, or can be judged by three methods. So let us put the heading as screening of transformed cells. So how would you screen them? How can you screen the transformed cells? You can screen the transformed cells by two methods. First one is antibiotic resistant gene, and second one would be the blue white screening method, or enzyme mediated. So first of all, let us understand the first one, antibiotic resistance gene. So this is the vector that we had taken. It already has, the vector has multiple genes. It has ORI, that is origin of replication. It also has genes that are responsible for providing resistance, such as ampicillin resistant gene, such as tetracycline resistant genes. Let us take these two genes for our example. Now you also have your gene of interest over here. Okay, now when you make these this recombinant DNA to enter into the E. coli. Suppose this is the E. coli cell, right? And what do? You make this recombinant DNA enter inside the this E. coli. Let me show you, just like this. Okay. So this is one such possibility, and multiple E. coli cells would be present over there. Multiple E. coli cells would be there. Suppose this is another E. coli cell, this is another E. coli cell, this is another E. coli cell, and it is not necessary that all of them would have been transformed. Suppose only one of them has been transformed, and all the others, suppose this is the first one, this is the second one, this is the third, fourth and fifth. Amongst 5 of them, only this one, the first one, has got the ampicillin resistance gene, and the place where all these E. coli cells are grown, the media has been provided with the ampicillin resistant the ampicillin drug, right? So the E. coli, only that E. coli which has the ampicillin resistance gene would survive in these conditions, and all the other E. coli would be dead. Okay, so all the other would be dead. This would die, this would die, this would die, this would die. Why? Because only this has been transformed and all the others were not able to be transformed. So this was how the antibiotic resistance gene can be used for screening, right? Now let us understand the screening through the enzyme method. Screening through enzyme method. Okay, so let me choose a color. Screening through enzyme, right? Suppose this is the vector, right? And in this particular vector there is a gene. Okay, let me change the color for your understanding. Here is a gene, right? Here is a gene. The name of this gene is LAC-Z. Okay, the function of this LAC-Z is to form beta galactosidase. So beta galactosidase is an enzyme that can convert X-gal into a blue colored product, right? So this X-gal, what is this X-gal and where is it found? How come this X-gal came into the picture suddenly? This X-gal is present in that particular medium in which these E. coli are growing, right? Let us understand this. So we had said that the E. coli, we are putting these recombinant cells into E. coli. We have already said that this LAC-Z gene is present over here, and LAC-Z gene synthesizes or produces, what does it produce? It produces beta galactosidase. We have already said that the function of beta galactosidase is to convert the X-gal into blue colored product, right? So in this what do we need to understand? This is a complete vector. So when you insert your gene of interest in here, the gene of interest is essentially inserted into this red color portion. Okay, so let me show you. Let me make you understand with a diagram. Okay, so this is your gene of interest. This blue color fragment is showing your gene of interest. The red color is showing the LAC-Z gene. The red color is showing the LAC-Z gene. Okay, the function of LAC-Z gene I have already told you, for synthesis of beta galactosidase. Now if you look closely, then you would see that this gene of interest is basically making this LAC-Z gene non-functional. Why non-functional? Because it is getting attached in between the LAC-Z gene, so the LAC-Z gene would not be able to make beta galactosidase if the gene of interest has been attached here. Okay, let us understand this again. If the gene of interest has been integrated into this vector correctly, then the beta galactosidase would not be formed. Okay, if gene of interest is not integrated, then beta galactosidase would be formed. Okay, so this would be clear to you. I guess this would have been clear to you. So now if you again make the E. coli cells, if you again make the E. coli cells, so these are the E. coli cells that we have been working upon. These are the E. coli cells, and in some places, in some cases, suppose this is integrated into this first E. coli, and not integrated into the second, third and fourth E. coli. Okay, so what will happen according to you? Okay, in the media, in the petri dish in which these E. coli are growing, X-gal is present. In the media, X-gal is present. So in the media, X-gal is present, so the X-gal would be utilized by these bacteria, and only where the gene is intact, only where the transformation has not taken place, or only where the gene of interest has not been attached to the vector, only at that places the blue color would appear in the colonies. And for those places at which the gene of interest has properly integrated into this vector and transformation has taken place, then what colored colony would be present there? Blue colored colony, white colored colony would be present over here. White colored colony. Why white colored colony would be present over here? Because white colored colony would be present because here gene of interest has been integrated between the LAC-Z gene, and LAC-Z gene would here be non-functional, and therefore it won't be able to produce the beta galactosidase, and when beta galactosidase would not be able to be produced, it won't convert the X-gal into the blue colored product. Therefore the colonies would appear white, whereas in contradiction, if the gene of interest is not present here, it is not transformed, the vector it has not been properly integrated into the vector, and no transformation has been shown, then what will happen is the gene of interest, the LAC-Z gene would be intact, no gene of interest would be present, and due to the presence of LAC-Z gene, beta galactosidase would produce, and finally the X-gal would be converted into your X-gal would be converted into blue colored, and therefore blue colored colonies would appear. So this was all about the recombinant DNA technology and screening also through antibiotic resistance gene and blue white screening. I hope this video would have been useful to you, and if you have liked this video, please click on the like button and subscribe to my channel. If you have any queries related to this, please type it in the comment box and I would resolve it in the upcoming lectures. Thank you so much.